Let me share my screen first. If you can see the slides, please let me know in the chat box and then I will start. If the slides are visible to all of you, then please, somebody please confirm in the chat box and then I will start. If the slides are visible, yes, okay, fine. Thank you for the confirmation. So in the last session, we ended with this question number 6 and let us begin with the next question one by one. This question will be very important because in this session we'll be learning to identify some peptide sequences as well through by the cleavage of the peptides bond, and then identifying which enzyme is cleaving which bonds. We'll be able to identify the sequence as well. So we'll be doing or solving those kind of problems. So I would expect all of you to try it by yourself and then I will discuss the results as well. Let me start. Fine. This question was finished. So we will start from question number 7 in this session. So the first question states that if the given diagram is a reaction scheme of Edman degradation, what is A? This is your question. Okay. So these are the options. So this is a very easy one. Basically they are asking what is the reagent that is being used in case of Edman degradation. I feel that all of you know this answer. So please write your answers in the chat box and then I will show you the correct answer. So there are 4 options: phenyl isothiocyanate, B acetic acid, C is FDNB and D is nylon 6,6. So these are the 4 options, and in the right hand side the scheme of Edman degradation is shown. So you have to identify what is A. Okay, fine. Still I did not get any answer. So what do we do in case of Edman degradation? So through Edman degradation we are basically will be able to understand what is the end group or the end side group, end terminal group of a peptide chain. So we all know in case of any peptide we have our N terminal and have our C terminal. In case of Edman degradation, it is useful because it will tell us that what amino acid is sitting at the N terminal. This N terminal amino acid will come out and then will generate another shorter peptide version. And after this side of amino acid cleaves, obviously there will be generation of a new N terminal, because one of the amino acid which was previously at the N terminal is now cleaved, and now you are ended up with a new peptide or a shorter peptide with a new N terminal. And in case of Edman degradation, this process will continue throughout. Whatever is the N terminal, whoever is sitting here, one by one will be cleaved out. In the next step, N prime will be cleaved out and you will be ending up with something like this, where there is a new N terminal. So this is the whole process. Now the question was, what is the reagent that is used in case of Edman degradation? Now if you do remember, the reagent that is used in case, this is written as PITC, or known as phenyl isothiocyanate. It looks something like this, phenyl isothiocyanate. Okay, and this is after this phenyl isothiocyanate, you can see that this NH2 or N terminal amino acids NH2 reacts with this carbon center and form this particular molecule, which is otherwise known as PTC or phenyl thiocarbamyl moiety. Okay, and after that phenyl thiohydrantoin will be removed from the system and you will be ended up with a shorter chain of the peptide molecule that you are given. So the correct answer for question number 7 should have been the reagent that is used in case of Edman degradation is phenyl isothiocyanate, otherwise known as PITC. So our correct option for this question should have been option number A. Okay. Moving to next one. Now read this carefully. The questions can be a bit long. You have to read this with patience, but the questions are actually very easy. The words are just there to confuse you a bit. Okay. So in this question number 8, it states heme is a prosthetic group of myoglobin and hemoglobin. Fine. Y is the organic portion of the heme molecule. So we have a heme molecule. Okay. Y is the prosthetic group. One second. Y is the organic portion of heme molecule. Fine. The only difference between Y and the heme moiety is the presence of a tetra-coordinated divalent M atom. So it states that this heme moiety consisted of a metal ion that is written as M and another part that is given as Y, that is the organic part. Okay. Again it states M is extremely important for binding oxygen and hence plays an important role in its transport and storage. So you basically need to identify Y and M. So Y is the organic part. So basically you are dealing with a holoenzyme where you have an organic part and another non-organic part or a prosthetic group basically. So your prosthetic group is a metal atom that is very essential for binding oxygen molecule, and there is another organic part that is giving the importance to this metal ion for all the reactions that is happening. So you have to identify what is the metal ion and what is the organic part. So we all know what is heme. It is basically talking about hemoglobin. So what is the role of heme in case of the M or here heme part or the metal part in case of hemoglobin is, we all know, Fe2 plus. So our metal is in this case is Fe2 plus, and what is the organic part? It is the porphyrin rings, which is otherwise known as protoporphyrin 9. Okay, so that is your outside where the 4 porphyrin moieties are attached together. It is called protoporphyrin 9, and in the center of that protoporphyrin there sits Fe2 plus, which helps in binding the oxygen molecules. So our correct answer should be that Y is protoporphyrin 9 and M is Fe2 plus. So the correct answer of this question should be option number C. Fine. Okay. So this was actually, there is the structure of the heme molecule overall, but I have written over it. So if you still look, this is the overall protoporphyrin 9. This is the whole organic part, and there sits your Fe2 plus. Okay. Moving to the next one. This is again very important. Chymotrypsin will cleave on the carboxyl side of which of the following groups of amino acids in a protein molecule? So you have a serine protease, one type of serine, example of serine protease, that is chymotrypsin. So it is asking that it does cleave on the carboxyl side of some certain side groups of amino acids. Which amino acids will it choose in case of a protein molecule? Now if you do remember, in your class it was clearly mentioned that in case of chymotrypsin, where does it sit? Which of the molecules does it prefer? If you, I still do not see any of the option. It's just a memory based question, that what kind of amino acid residues does chymotrypsin prefer? Just a general question. If you remember about the whole mechanism and everything, you will be able to answer that. If anybody can answer this, please write it down. Otherwise, I will obviously show you the correct option. Anyway, fine. So, chymotrypsin basically cleaves the carboxyl sites of hydrophobic side chain groups, amino acids that have hydrophobic side chains. So, among these options, only phenylalanine, tyrosine and tryptophan, these are the amino acids. If you do remember their structure, all of them have a aromatic ring in their side chain, like pH, phenylalanine, in that case CH2pH, tyrosine, that means CH2pH, like that. So chymotrypsin will always prefer amino acids those who have a hydrophobic side groups or a bulky hydrophobic side chains, and then chymotrypsin will go there and cleave the carboxylic side of those particular amino acids. Okay, you do need to remember this kind of information, because whenever we will be solving the peptide sequences, you will need to remember that what enzyme acts upon which kind of amino acids. If you do not know, you will not be able to identify the peptide sequence or form the peptide sequence in that case. Okay, so you do require to remember their role, like different different enzymes like chymotrypsin, trypsin, esterases, or some other organic molecules like CNBR, etc. You need to remember those things, because otherwise it will be difficult for you to solve the overall problem. So for now, we know that chymotrypsin will always choose hydrophobic, bulky hydrophobic amino acid residues. Fine. Remember this information, because we will be needing this again in some question. Okay. Coming to the next question. Again, it is a big question, but read it very carefully, line by line, and you will be able to answer this. So it states, a peptide is generally hydrolyzed into its constituent amino acid by treating with 6-normal HCl at 110 degrees C for 24 hours. So basically, you are separating down or cleaving the peptide bonds and getting the constituent amino acids by treating with concentrated HCl. The amino acids that are obtained in the hydrolyzed states are chromatographically separated. So now your peptide bond, see, suppose this was your peptide bond, you have cleaved all the amino acid by using 6-normal HCl and boiling it to 110 degrees C. And say suppose now you have different residues. You need to separate them. So those amino acids are separated chromatographically. Fine. Now you need to analyze quantitatively that which amino acids are there, how much amount of those amino acids are there. Okay, for that what we are doing, they are saying that they are quantitatively analyzed by reaction with C. Upon treating the amino acids with C, an intense blue color is obtained. But the amino acid D does not give a blue color. It gives a yellow color due to the structural uniqueness compared to the other amino acids. You have to identify C and D respectively. So the big question in short, that suppose you have these different type of amino acids. You can name them instead of 1, 2, 3, 4. Suppose we are naming them A, B, D, E, F, G, etc. These are the amino acids. You do not know, you have labeled them. Now you are trying to quantify them. In all of them, after separating them, you have added a reagent that is known as C. You have to identify C. Most of the amino acids are giving you blue coloration except this amino acid D. This is not giving blue coloration, instead it is giving yellow coloration. So you need to identify what is the reagent used, that is C, and what is that amino acid which was giving yellow color instead of blue. So let us look at our option. These are the 4 options. First of all, just by reading the last line, you should already understand that there is only one compound that is used for blue coloration in case of amino acid, and that is your ninhydrin. We have already read this multiple times that ninhydrin actually interacts with these amino acids and gives you a blue coloration. There is only one amino acid, so we already know that the reagent C that we were talking about is nothing but ninhydrin. We have already understood this. Fine. Then the question comes that what amino acid is D, which is not giving blue color but yellow. So there is only one amino acid which does this kind of problem. This is not a problem, rather it is kind of a easier identification for this amino acid. It has a certain structure because of which it only can give you a yellow coloration. I will come to that. And this structural variation only exists in case of proline. Why? I am showing you. One second. This is the mechanism is not there. One second. I think I should have the mechanism somewhere. Ninhydrin. Just give me a second. Okay, fine. I will come to this question later as well. This is the same question. Let us look at the reaction that ninhydrin has with the amino acids and gives you the blue color. So this is, one second, this is your ninhydrin. It is interacting with your amino acid here. This is the whole mechanism. One second. Yes, this is the entire mechanism. So this is your ninhydrin and this is your amino acid. And via this mechanism, the entire reaction is happening and ending up with this end product which is purple in color. So all the amino acids that have this NH2 group can react with ninhydrin and end up with this purple colored anion. But there is only one amino acid, that is proline, which does not have a NH2 group. It has a NH group in his ring. It is not a primary amine but a secondary amine. Thus, if you think about it, in case of proline, because you know in case of proline there is only a one hydrogen atom. So, after one attack and removing one hydrogen atom, there is no more hydrogen atom left to carry forward this reaction through this step. Because you can see in the next step there is the breaking of another NH bond. For proline it is not available, it only has one NH bond. So in that case, once the one NH bond has already reacted here, it has no other NH bond in expense to do the next steps. So the reaction stops at this stage only. It cannot follow through all the steps to go to the purple anion. That's why only in case of proline in ninhydrin test we end up with a yellow coloration. It somewhat looks like this. Now it will be very easy for you to understand. Whenever there is a primary amine group, it can react with ninhydrin and give you this purple coloration. It is extremely dark, that's why it is looking like black. It is an extremely dark blue or dark purple color. Okay. But in case of proline, once this NH is already interacting here, after that it has no more hydrogen for expense. That's why the reaction stops here and you end up with a coloration like this. So back to our original question. One second. So we are solving this one. Yes. So back to our original question is our D amino acid marked as D, which was not giving blue but a yellow color, is nothing but basically proline. So we can write it down here. So our D was proline. So what should be the correct option then? That our reagent used C was ninhydrin and D was proline. So the correct option for this question should be option number C. Okay. Moving to the next question. It is stating A is an enzyme that cleaves the phosphodiester bond between nucleotides of RNA and plays a diverse range of roles in RNA metabolism. It is generally secreted from pancreas. What is A? So again they are talking about an enzyme A which breaks the phosphodiester bonds that exist in case of RNA. We all know that it is very yes, I got one answer now. Oh, sorry, yes. I think the answers came late. Yes, for this question you already, all of you know the answer. For RNA, the enzyme that is used for cleaving is basically ribonuclease. So yes, the correct option for question number 11 is C, ribonuclease. Okay, so let us move to the next one. So this is your next question where it states, from the given graph, it can be said that at lower pH, hemoglobin's affinity for oxygen molecule is what? Whether it increases, decreases, remains same, or it does depend on the blood type. So what it is talking? At lower pH, what happens to the affinity of hemoglobin for oxygen? So the graph is already shown on the right. If anybody do know the answer, please do write it. I will obviously explain. Just look at the graph and you will be able to tell it. What happens to hemoglobin at lower pH in terms of affinity towards oxygen molecules? Okay. If we do look at the graph, you can already tell. This is the graph for pH 7.4 and this green one is for pH 7.2. So what is happening when you are lowering the pH? If you look, suppose at this partial pressure we are checking. This is partial pressure of oxygen. So suppose it is some value where we are checking the affinity of hemoglobin for oxygen. So let us go here. Let us see. For the green one, this is the fractional saturation of oxygen at pH 7.2 at this partial pressure of oxygen. And what is for the red curve? That is pH 7.4. This will go a bit more up. And for pH 7.4, this is your fractional saturation. So obviously what we can see? At pH 7.4, that is the red graph, at same partial pressure of oxygen, the fractional saturation is much higher than that of pH 7.2. Just by drawing the lines, we can very easily understand that. And that is very necessary as well, because we already know that in case of our muscles, tissues, the pH is lower. And whenever the pH gets low, the affinity of hemoglobin for oxygen molecule decreases. It has to, because only in that case it will give away the oxygen molecule and myoglobins that are already sitting at the tissue site, they will take the oxygens from hemoglobin. If hemoglobin's affinity does not decrease at lower pH when it going to muscles, then it will never transfer the oxygen molecules to myoglobin. So obviously at a lower pH, the hemoglobin's affinity for oxygen has to decrease. That is the biological significance. And from mathematically from the graph, it is very much you can see from the lines as well easily. Okay, yes, the correct option that you wrote, Akshara, I guess, yes, the correct option then for question number 12 should be option number B. Okay, yes, that at lower pH hemoglobin's affinity for oxygen molecules decreases. Fine. Moving to the next one. It is stating strychnine is a convulsant poison. Fine. Then they are saying glycine is a major postsynaptic inhibitory neurotransmitter in mammalian spinal cord and brainstem. You do not need to remember all these words. Just need to focus that glycine is something that is very important in our mammalian spinal cord and brainstem. Fine. But strychnine is a poison. So what do we have? Here is our suppose strychnine. We know this is a poison and we know that glycine is very important in our mammalian spinal cord and brainstem. Fine. The next line states strychnine binds at a separate binding site on the glycine receptor in such a manner that its binding lowers the affinity of glycine receptor for glycine. So what does this statement means? First of all, if glycine is very important, then glycine needs to bind somewhere. Then that means glycine will have some receptors in our body where glycine molecules can go and attach, right? So glycine should have some receptor molecules. Same like enzyme substrate complexes, same thing. So suppose our receptor looks something like this, and this is our glycine molecule which fits nicely in the cavity and does its job, whatever it is. But in the question it is saying strychnine, the poison, binds separate binding site on the glycine receptor. So it is the glycine receptor, but strychnine binds at a separate binding site. So we already know that some poisons or inhibitors not always target the active or catalytic site. So this is the site for binding for glycine. Fine. But sometimes some inhibitors or poisons does not target the catalytic site but some other site. Suppose it binds here. Fine. That is what they said. Some other binding site than the glycine receptor. After it binds, it says, thus strychnine hinders the action of an inhibitory transmitter leading to convulsion. That means after it binds, it somehow hampers the binding of glycine to the receptors. So how can it be done? It might look something like that, that after the poison binds, then the receptors say somehow start to look like this, and the glycine molecule say now the active site looks something like this. Now the glycine molecule, here is the poison sitting, can no longer attach to this receptor. So this is the whole scenario that is going on. So what can we call this strychnine molecule for glycine receptor? What kind of reagent we will call strychnine for glycine receptor? So if you do remember, where we talked about different kind of inhibition, there is only one example under which the inhibitor attaches to the enzyme and makes some conformational change in the enzyme, which ultimately results in the conformational variation of the catalytic site, and it can no longer bind to the substrate. And this only happens, yes, absolutely right, it only happens, Shishti, yes, right, it only happens in case of allosteric inhibition. where there is a conformational change in your enzyme and your substrate can no longer bind. So this is the whole scenario that was going on for strychnine. So what is strychnine for our case? It is nothing but an allosteric inhibitor. Okay. So see, it is a very easy question. You already know what is allosteric inhibition. What just you need to know in this question or do, that is read the lines very carefully and understand what they are saying. And then it is very easy for all of you to answer. Okay. Fine. Moving to next one. Again, similar question. This is also a very easy question, but it looks like a very big one and confusing one. So let us read one by one again. So it states, at higher altitudes or in mountain terrains, oxygen's availability is generally less. It is very obvious. In such cases, the oxygen saturation in our body is increased by releasing the oxygen bound to hemoglobin. This release of oxygen in our bloodstream is helped by a compound called X. Okay. X is produced in order to bind with 2-deoxyhemoglobin moieties, thereby releasing the bound oxygen in the bloodstream. Again it states X has a very high affinity to our deoxyhemoglobin and binds very strongly with it. The question is very long and actually the answer is very easy. I already have one answer, I can see. So basically it is stating that you have your 4 subunits of hemoglobin, but whenever you are, yes, I am getting many answers, good. So whenever you are going at mountain region, the oxygen molecules that were binding, because the oxygen quantity is now low, so there will be a less amount of oxygen in your body. So obviously that will be a problematic situation for you. Now the role of X comes to play. What does it do? It releases the oxygen molecule, okay, and say suppose binds in the subunit. Fine. It binds on the subunits of hemoglobin. That means one molecule of oxygen is now released. That means the oxygen requirement in your body is fulfilled in this way. Although outside the oxygen amount is low, but due to this compound X, the required amount that you need is already fulfilled because of the release of O2, and that only happens because of binding by X. So this is nothing but the compound that is itself produced by hemoglobins, otherwise known as BPG or 2,3-biphosphoglycerate. Yes, the answer was already given for many of you. Very good. So this was our correct answer. Okay. This is again very important question. I would like all of you to solve this particular one individually. It states, upon performing an experiment, a student obtains the following peptide residue. So you have your peptide residue given to you. The student then does the following experiment on the residue separately. So this is your peptide residue. Now you are performing this experiment on this peptide sequence. The first one is Edman degradation. Fine. The second one is treatment with trypsin. And the third one is treatment with cyanogen bromide. So you are doing these experiments one by one. So you take this peptide and first you perform Edman degradation. Again you take this peptide and you treat with trypsin. Again you take this peptide and treat with CNBR. What will you obtain in each of these experiment? I would like to, for all of you, to answer this, write it down. Okay, you can write in the short letter form as well. Instead of this 3-letter code, you can use the 1-letter code as well. But please do try it once by yourself. I will give you 2 minutes. You can take the help of the lecture material as well. You already know what does Edman degradation do in this session as well. I just told all of you that what does Edman degradation do. You can take the help of the lecture material to remember what is the role for trypsin and what is the role of CNBR, where do they break the peptide bonds, and then write the answers. Okay, please tell me the first answer, at least for Edman degradation. 2. Okay, I do not quite understand what does 2 mean. Okay. For your help, you can look at the options as well. Okay. Please do write the peptide sequence. You are choosing option 2. Okay, fine. I haven't shown you the option. These are your options. Okay. First, write down the peptide sequence in your notebook. Okay. Do write the peptide sequence and also do write the 3 experiments. And then I will change the slide and you can look at the option and choose the correct option from there. First, please note down the peptide sequence. Let me note it down as well. Fine. And these are your options. First one is Edman degradation, second one is use of trypsin, and third one is use of CNBR. Fine. So let us see what should we get. Let me write the 1 second. First let me write down the sequence. So your sequence was threonine, phenylalanine, valine, lysine, alanine, tryptophan, glycine, methionine. So this was your peptide sequence. I have one answer. Very good. So yes, the answer that you gave, Shrishti, is correct. Now let me discuss this. So our first experiment that was performed was Edman degradation. So all know what does Edman degradation does. It cleaves the amino acid that is sitting on the N terminal. So this is your peptide bond. So already know this side, the left hand side should be N terminal and this side should be your methionine terminal. Okay, the peptide sequence is joined like that. I just could not get the whole space, that's why. So this is your N terminal and this is your C terminal. So obviously in case of Edman degradation, it will cleave down the amino acid that is sitting down at the N terminal. So after Edman degradation, in one part you will get threonine and the rest of the peptide will be sitting there accurately. Just 1 mole of Edman degradation reagent has been used and 1 threonine has been clipped. Okay. The next thing that has been performed is the use of trypsin. Now where does trypsin breaks the peptide bonds? It breaks after, or it breaks at the carboxyl site of arginine and lysine. So again you need to remember that what is the role of trypsin. So trypsin cuts after arginine and lysine at the carboxylic site. So in your entire peptide sequence, if you look, you do not have any arginine, but you have one lysine amino acid residue I can see here. So trypsin will cut after lysine, that means at the carboxylic site. Fine. So if it does cut at the carboxylic site, now you are left with this portion, that is threonine, phenylalanine, valine, lysine. So this is your portion, THR, phe, val, lys. Okay, fine. And another part is, after cleaving at this portion, your another part is alanine, alanine, tryptophan, glycine, methionine. Okay, and this is your part. So this is correct. Moving to the last option, that is CNBR. Again also here you have to remember what does CNBR do in case of peptide bonds. So CNBR cleaves after methionine residues at the carboxyl site. So if you do remember, this is your entire sequence and there is only one methionine. Now CNBR cleaves after methionine at the carboxylic site. So this should be this position, after methionine carboxylic site. But you do not, your entire peptide sequence is finished at methionine. Methionine itself is sitting on the carboxylic site. There is no other residue after methionine. So there is nothing for CNBR to cleave there. If there is suppose another amino acid residue, if there was present like X after methionine, then CNBR would have cut the peptide sequence there. But as it does not have any other sequence after methionine, so that means there is nothing for you to cleave. So the third option is that after treating with CNBR, the original peptide chain will remain because methionine is itself sitting at the carboxyl end of the peptide. Okay. Hope this is clear. Please do remember these things so that it will be easy for you in case of exam as well as assignments. Fine. This is the next question. It states that the catalytic triad of most serine proteases contains aspartic acid, histidine and serine residues. Which of the following is not true? So the word that is important for you is this particular one, that you need to choose the false one. Not true means you have to identify the false statements. These are your options. Let us come to this one by one. The first option states that the aspartic acid residue is used in an acid catalyzed attack on the peptide bond. Now if you do remember the mechanism of, say, one of the example of serine proteases like chymotrypsin, we have already read that in our lecture material, that what is the mechanism for chymotrypsin. So what is the triads that are used, catalytic triad that is used in case of chymotrypsin? Say aspartic acid 102, histidine 57 and serine 195. Now if you do remember the mechanism of chymotrypsin, there aspartic acid only had one role, that is to set the histidine or to change the fix the histidine in a particular position that is accurate for the enzyme, entire enzyme to act. So basically aspartic acid fix the position of histidine in a particular manner so that the other following steps can take place. So aspartic acid as such has no direct role in case of chymotrypsin or serine proteases type of enzymes. It only has one role, that is to fix the position of histidine. Okay, so the first statement which states that aspartic acid is actually in a acid catalyzed attack is wrong. Basically it does not have any active participation in the whole enzymatic process. So the first statement is obviously wrong. So we have identified our first non-true statement. Okay. The next thing, the histidine residue is responsible for the substrate specificity of trypsin. Again here as well, histidine residue is responsible for substrate specificity. Now if you do remember, if we look at the enzyme, if this is the active site where serine sits, then there are another two sites. Let me just this side is known for as oxyanion hole and this side is known as hydrophobic pocket. If you look at the mechanism of trypsin, you will understand that after the oxyanion formation, that needs to be stabilized in some way. There is a oxyanion formation in case of trypsin and that needs to be stabilized in some way. For that, enzyme has a specific place where some residues like serine and glycine sits and stabilize the oxyanion via hydrogen bond formation. Okay, but it is not your catalytic site. This is your catalytic site where serine is doing all the work. But for the substrate, once the oxyanion has formed, it needs to be stabilized by some amount as well. Again, for chymotrypsin, the substrate it chooses is the amino acid residue with hydrophobic side chains. So while the reaction is going on, you also need to somehow stabilize the hydrophobic side chain in the interaction with the enzyme. For that, the enzyme also have a hydrophobic pocket where the amino acid hydrophobic side chain can be stabilized, sit and stabilize. So the catalyst or the enzymes actually has all the thing. It has the active site that is necessary for reaction. It has the another site that is necessary to stabilize the transition states, and there is another site which is necessary for stabilizing the side chains. So overall these three centers all together is responsible for substrate specificity. Any of these 3 points is missing, then the substrate won't work properly for the enzyme. So for substrate specificity, all these 3 points needs to be considered. So where it is stated that histidine residue is only one that is responsible substrate specificity is absolutely wrong. The entire substrate specificity depends on many amino acids, like where serine will attack, what are the hydrophobic groups that are present in the enzyme, and like other moieties like glycine and serine, which form hydrogen bonds with the oxygen ion, they are all important. So our next statement as well is a false one. Let's look at the third one. It states, yeah, I already have one answer. Yes, fine. The third statement says the serine residue is used in a nucleophilic attack on the peptide bond, which is very true if you look at the mechanism of chymotrypsin. In case of serine, it forms O minus and then attack this carbonyl center, and we get a transition from sp2 carbon to sp3 carbon. So the third statement is actually a true statement. Coming to the last one, it says the aspartic acid residue is responsible for the substrate specificity of trypsin. Again, just I said, any of the one amino acid residue will not be sufficient for ensuing the substrate specificity. All of the factors have to be taken care of for substrate specificity. So again, the fourth option is similarly a false statement like statement 2. So the only option true was statement number 3. Now in the question we had to select the false ones. So our 1, 2 and 4 are false option. So yes, Manisha, yes, you are correct. The option that should we choose is option number C, that 1, 2 and 4 are not true. They are the false option. No, the B will, this is the thing. You who opt for the option B, you chose the correct option. But in the question, it is very clearly mentioned that which of the following is not true. So you had to choose the false ones. So this is, you have to be very careful while answering, because you might be that you know the answer, but because of the how the question is written, that's why you might have selected the wrong one. So you have to be very careful while answering this question. So the correct option should be option number C. Okay. Moving to next one, it says identify A, B, C and D from the graph shown below. This is the graph of percentage of saturation versus partial pressure of oxygen. Okay. You have to choose what is A, B, C, D. A is the red graph, B is the black graph, C is the pink bar graph and D is the green colored bar graph. You have to choose what is A, B, C, D. I think all of you know this. Yes, I have already one answer. Yes, and it is absolutely correct. The correct option for this is option B. Just look at the nature of the curve. The nature of this red line is sigmoidal. That means the only one who shows sigmoidal fashion of saturation is hemoglobin. In case of myoglobin, it is hyperbolic. It is, we all know, in case of myoglobin, as there is no cooperation effect, so it shows a hyperbolic curve in case of saturation. Okay. And see this pink bar graph. Here in the pink bar graph, the partial pressure of oxygen is very low. Suppose in the range of like 30. That means where is the partial pressure low in our body? In case of tissues. So the pink one, or where the partial pressure is low, should be the region for tissues. And the green one, we can see the partial pressure is 100, full saturation. And where do we have the full saturation of oxygen? Obviously in case of our lungs. So D should be our lungs, that is the partial pressure in our lungs. Yes, both of you are correct. Option number B was the correct option. Coming to next question. Question number 18, it states that identify the correct representation of Bohr effect in association with the process of oxygen transfer between the lungs and the muscle by hemoglobin. So this is a very one, I just quick easy one, I will quickly show you the answer. So what happens? The correct option is option A. Why so? Because we have our oxyhemoglobin. Oxyhemoglobin, where does it coming from? It is coming from lungs. Obviously we know in lungs there is very high amount of oxygen. So hemoglobin will bind to oxygen and come as oxyhemoglobin. Now it is going to the muscles. Oxyhemoglobin is going to the muscle, and as soon as the pH is lowered there, it will start releasing oxygen in exchange of H plus and CO2. It will instead attach with H plus and CO2 and start releasing oxygen molecules. So obviously this particular representation was correct. This is incorrect because obviously from muscle you cannot form oxyhemoglobin. In muscle oxyhemoglobin dissociates to hemoglobin. So this was again wrong. These representations were wrong because when the hemoglobin releases oxygen molecule, it immediately binds to H plus and CO2. So it is basically in expense of H plus and CO2, it releases the oxygen molecules. So this representation that they are all separately existing is wrong. Here also this representation is wrong. Hence both these options are incorrect. Moving to the next one. Okay, I just said this, I will quickly go through it. Chymotrypsin selectively cleaves at the, we already know, at the carboxyl site of bulky hydrophobic amino acids. So our correct option should be option number D. Chymotrypsin selectively cleaves at the carboxyl site of hydrophobic amino acids. Fine. Moving to next one. It states in Edman degradation, phenyl isothiocyanate reacts with terminal dash groups. So we already went through Edman degradation. We know what is used in case of Edman degradation. That is phenyl isothiocyanate, PHNCS. Now this reacts with always the N terminal of your peptide chain. So the first option should be, what should be the option? You should, and it interacts with the N terminal and breaks down the peptide bond at the what side? Amino site. N terminal means amino site. So at the N terminal or amino site, it breaks the peptides and the first reaction happens under slightly alkaline condition. If you look at the mechanism, I think I showed it in the last session. If you look at the mechanism, then you will see that at the first step the reaction happens at a slightly alkaline medium. After that it forms phenyl thiocarbamyl derivatives, and at the last, that is after that, it forms phenyl thiocarbamyl derivatives, from which we end up with phenyl thiohydrantoins, and this reaction happens in presence of H plus or a mildly acidic condition. So at the end it should be that phenyl thiohydrantoin is liberated under mildly acidic conditions. So this is the whole statement. In Edman degradation, phenyl isothiocyanate reacts with terminal amino groups of peptides under mildly basic conditions to form phenyl thiocarbamyl derivatives, and the modified cyclic derivative of terminal amino acids is liberated as phenyl thiohydrantoin under mildly acidic conditions. Okay. Moving to the next one. This also you know already. The molecule shown here coordinates with the metal ion M2 plus for their functioning in the human blood. When X coordinates with M, this structure is referred as Y. M and Y are what? So you already know what is M, what sits at the centers. this is Fe2 plus. In case of hemoglobin, you already know this is Fe2 plus. And what is Y? That is the organic part. That is basically your overall. That should be, I think it is written as this. Excuse me, someone's mic is on. If you need to say something, you can just write it. Okay. When this entire structure, so this organic part is, they are stating it as X, or X is your porphyrin. Someone's mic is on, please. If you have any query, please to write in the chat box. I will explain. Okay, I am seeing it, so please write it down. Do not turn on your mics. So coming back to it, so your metal is Fe2 plus. The organic portion is porphyrin, this X, which is denoted in this question as X, and together Fe2 plus plus porphyrin, together we will call it heme. So our M is Fe and together Y is heme. We already know this. So moving to the next one. Okay, so this question is already discussed. I will not be discussing. It is the same one, that at higher altitudes, that at mountains, hemoglobin binds to a molecule called Z. And what is that? You have already answered this, that it is 2,3-biphosphoglycerate, and it binds to hemoglobin at higher altitudes so that it can minimize the oxygen requirement in oxygen deficient areas, because as it is binding with your hemoglobin, it is releasing the oxygen, so the requirement, the amount that you require to intake from outside is now becoming less. So this is their job, so I am not spending much time in that. This is the next question. It states, okay, this is a very interesting one. You have to form the peptide sequence. So it states a certain peptide fragment is treated with Edmond reagent, CNBR and carboxypeptidase separately. So in this case, you have to identify the overall peptide. So you don't know what is the overall sequence of your peptide, which one is the N terminal, which is the C terminal, you don't know. But when you are performing Edmond degradation, so first we have our Edmond degradation. It is giving you valine. Okay. When you are treating with CNBR, that is giving you VGA. Again, fine. VGA lactone. Sorry, that should be mentioned. VGA lactone. And the final one, and VGA lactone plus LSPR, that you are getting from CNBR treatment. And on the other hand, treatment with carboxypeptidase yields R. So whenever you are using carboxypeptidase, you are getting R. Okay. So these are the residues that you are getting by these treatments. Now you have to identify what was your peptide fragment at the beginning. Okay, this is a very interesting one. Try to solve it by yourself. You just need to remember, what does Edmond degradation do, or Edmond reagent do? What do you get after Edmond reagent? Treating with Edmond reagent. What do you get after treating with CNBR reagent, and so as carboxypeptidase? Once you remember those or identify those, forming the peptide sequence will be a piece of cake. Okay. Please try it out by yourself. I will give you all 2 minutes and then I will show you. For your help, I am also showing you the options. It might get easier for all of you to now choose one of those options. Please try it yourselves. It is a very easy one. Just need to remember the role of all those reagents. I have one answer. Okay, very good. Okay, so let us see. Yes, absolutely the correct option that Shrishti said, it is correct. The correct option is option number D. So how she solved it? Let us look at it. 1 second, yeah. Coming to the first statement, that Edman degradation gives you V or valine. So you already know what does Edman reagent does. If this is your peptide bond, it will straight go to the N terminal and cleave the amino acid residue that is sitting at the N terminal, right? Basically, so Edman degradation will always tell you what is the amino acid residue that is sitting at the N terminal. So from that, as we are getting valine, so we already know that valine is sitting at our N terminal. We already got that. It is our N terminal and valine is sitting there. So it is already known. Now the next part. CNBR is giving you two portions. After CNBR treatment, one part you are getting VGA lactone, another part you are getting LSPR. First of all, as you can already see, in one part you are getting VGA lactone. That means after V you can already see G is sitting after V and A is sitting after G. This sequence you can already see. From the first treatment or Edman reagent treatment, you already knew V was at the terminal. Now after CNBR, you are already getting that after V, there should be VGA. This matches with all the other treatments as well. This will give you V at the N terminal, and after CNBR, you are getting VGA. What is the other part that you are getting? Another part you are getting LSPR. So here again the sequence is intact. So you already know that after L comes S, then comes P and then comes R. You already know that. So this sequence is fixed. Now you need to remember that what does CNBR do? It does cleave after methionine residues at the carboxyl site. But in your sequence, you still haven't included methionine. In some part of the sequence, you have to add methionine so that CNBR can act and cleave it, right? So, now if we place our methionine residue here, now see if it does matches with the description of the segments that you are getting. If this is your sequence and now you treat it with CNBR, where it will cleave? It will cleave after methionine at the carboxyl site, and you will end up with this part and this part. This VGAM part will go into formation of VGA lactone, and another part will stay intact as LSPR. So it is exactly matching your definitions or the things that you are getting after enzymatic digestion. Let us come to the last point for confirmation. It says, on the other hand, treatment with carboxypeptidase yields are. What does carboxypeptidase gives you? It gives you an idea about your carboxy terminal amino acid residue. So whoever is sitting, whichever amino acid is sitting at the carboxyl end, carboxypeptidase will cleave that particular amino acid. Now it states that after CP treatment you are getting R. Now we have ended with this peptide sequence. If we treat with carboxypeptides, what will we get? Yes, we will get R because it will break at the carboxy terminal and separate the last amino acid that is sitting at the carboxy terminal. We will end up with R. So all the statements are now matching if our sequence was VGA MLPSR. So like that you have to solve or reconstruct the peptide sequences. This was a easy one. In some cases the sequence might be long and you might do many more enzymatic digestion to understand that and then solve it like a puzzle. Okay, hope it is clear to all of you. Moving to next one. I think this is the last question for these slides, I guess. So let us see. It is stating, given is the Hill plot for myoglobin and hemoglobin. Label the part marked as A, B, C and D accordingly to the states they corresponds to. So A, B, C and D are marked in the graph that is shown on the right hand side. You have to identify what is A, B, C and D. Okay, first try it out by yourself and then I will tell. I already have one answer. Okay. Okay, fine. So let us see. Yes, Akshara, you are absolutely correct. The correct option for this question should be option number D. So now let us look what they are saying. This graph, that is basically your percentage of saturation. This is your percentage of saturation versus log of partial pressure of oxygen. Now just look at the curve. We all know that in case of myoglobin only the saturation is fixed or log theta by 1 by theta is generally fixed. So if we plot log theta versus 1 by theta versus log of partial pressure for myoglobin, we do not see any kind of change in the slope. So our D has to be myoglobin because in that case we can see that there is no change in the slope. But in case of hemoglobin, we will always see a change of oxygen affinity with partial pressure of oxygen. How does it vary? So again one thing we can confirm that this red graph that is shown for A is nothing but hemoglobin because we can clearly see the change of slopes at different partial pressure of oxygen. Now coming to the different states. What happens when the partial pressure is very low? Here the partial pressure is 0 to 1. See, this is the extrapolation that is given as C. We can already tell that when the partial pressure is low for hemoglobin, this portion or saturation is also very low. That means in this particular condition of low pressure, hemoglobin is actually existing as a low affinity hemoglobin state. That means C in this low pressure region is existing as a low hemoglobin affinity state. But whenever you are increasing the partial pressure again, there is a sharp jump. Okay, and then when 4 of the sides are like fulfilled, again there is a saturation. Now this is extrapolated and marked as B. We can already see that when the pressure was high, the binding of value has also increased. That means B, the extrapolation of this portion, is actually the high affinity hemoglobin state. Now if we match all these things with our option, our correct option should be option D, where A is hemoglobin, B is high affinity hemoglobin state, C is low affinity hemoglobin state, and D is myoglobin. Okay, I think, oh no, we do have more questions. Fine. This states which among the following statements about myoglobin and hemoglobin are true. So what do we need to look for? We need to identify the true statements. Fine. The first statement is myoglobin is involved in oxygen transport and hemoglobin is involved in O2 storage. The complete opposite thing. We already know myoglobin is used for storage and hemoglobin is used for oxygen transport. So this first statement was false. Coming to the next one, hemoglobin has higher affinity of oxygen than myoglobin. Can it be true? Because if that was true, then hemoglobin will never release oxygen for tissues. We already know hemoglobins transport the oxygen molecules, and whenever it reaches the muscle and see the lower pH there, it releases the oxygen molecules, and because myoglobin's affinity is higher at that moment, it stores the oxygen molecules at the tissue site. So obviously, if hemoglobin always does have a higher affinity, it will never release the oxygen to the myoglobin. So our body will never function for the muscles. Okay. So again, this statement was false, that hemoglobin does not have a higher affinity of oxygen than myoglobin. This statement was false again. Coming to the third one, myoglobin binds oxygen in a hyperbolic fashion and each oxygen binds independently, and hemoglobin binds oxygen in a sigmoidal fashion and each oxygen binds cooperatively. Obviously, this statement is true. Why? Because we just saw the binding of myoglobin looks something like this and hemoglobins look something like this. This is partial pressure of O2, and if this is your saturation percentage, then the curves look like hyperbolic for myoglobin and sigmoidal for hemoglobin. Again, because myoglobin is a single unit, that's why the binding of oxygen molecule to each unit is independent. But for hemoglobin, as there are 4 subunits, that's why we can see a cooperative effect where when one oxygen molecule binds to one subunit, it promotes the binding of oxygen to other subunits as well. So the statement 3 was a true statement. Coming to the fourth one, it is saying hemoglobin is incapable of binding tightly with oxygen, whereas myoglobin is capable of binding tightly with oxygen. If hemoglobin cannot bind tightly with oxygen, then it will never be able to transfer the oxygen throughout the blood vessels, right? And then reach to the muscles. It has to bind. It has to bind tightly, but whenever it reaches in the muscles under certain conditions, it will release. It does not mean that it is incapable of binding tightly with the oxygen. Then in that case, it will no longer be of any use for transport. So again, the fourth option is false as well. So the only true option in our case was C. Yes, absolutely. Akshara, you chose the correct option. Option number 3 was the correct one. See, this already we discussed, this question about ninhydrin. Fine. This is also a very easy one, straight from the literature. So it is saying the protein ligand binding curve shown here looks something like this, where you use this equation y is equal to x divided by x plus z. You need to identify what is y, x and z. We already know y is the fraction of ligand bound. Suppose hemoglobin is binding to oxygen, so the y is our fraction of oxygen bound. X is the total ligand concentration, or say in this case total oxygen concentration, and Z is our dissociation constant or inverse of association constant, any of that. Either Z can be Kd, or in your, suppose in your question Ka is given, then in place of Z you can put Ka inverse as well. Okay, so Kd is just that at what concentration of ligand 50% of saturation is there is considered as the Kd. So it is straight from the literature. So let us move to the next one. I think we also know this. Which of the following allosteric regulators favor the unloading of oxygen in case of peripheral tissues? So what happens in case of tissues? In case of tissues, the hemoglobin unloads the oxygen molecules in expense of binding with H plus and CO2. We already know that. That means at the tissues you have high H plus concentration as well as high CO2 concentration. That means high H plus means high H plus and high CO2 concentration, because hemoglobin will release oxygen in expense of binding with H plus and CO2, the Bohr effect. So high H plus means low pH value and high CO2 means high partial pressure of CO2. Now let us see at the statements. Low pH, yes, absolutely low pH is required peripheral tissues for unloading of oxygen. Low 2,3 bisphosphoglycerate, that means BPG. No, if BPG concentration is low, then it will not bind to the deoxyhemoglobin and release the oxygen molecule. BPG's role is to bind with deoxyhemoglobin so that oxygen molecules cannot bind anymore, right? But if it is in low concentration, how will it regulate the unloading of oxygen? No, we require high concentration of BPG for unloading. So again, this is wrong. And the third option is low partial pressure. We already know the CO2 is high in concentration. So the partial pressure of CO2 should be high in our muscles. So this statement that low partial pressure is again wrong. So the only true statement is low pH. So the allosteric regulators that favor in this case for unloading of oxygen is low pH value. Let us look at the next one. Yes, fine. Which of the following statement is true? We already know this, that's why I am just showing it directly. Out of all those things, the first one states trypsin cleaves polypeptide on the amino acids of amino sites of arginine and lysines. This is wrong, because yes, it does breaks after arginine and lysine, but not the amino site. It breaks after the carboxyl sites. Okay. Next is elastase cleaves next to glycine and isoleucine. If you see, what does elastase do? It actually breaks after small hydrophobic amino acid residues. Okay, but glycine, isoleucine, these are not small hydrophobic, they do not have this small hydrophobic amino acid residues. So this is again wrong. So the next option is again wrong. Third statement is cyanogen bromide cleaves the peptide bond after methionine residues. Absolutely, we have done this already. This is correct. And the fourth option is chymotrypsin cleaves takes to hydrophilic groups. Absolutely wrong. Chymotrypsin breaks to the carboxylic site after bulky hydrophobic groups. We already know this, bulky hydrophobic groups and at the carboxyl site. We already know this. So our fourth option was wrong as well. So the only correct option was option number C. Okay, fine. Moving to the next one. Which among the following statement is false? Let us see. You have to identify the false statement. First one saying protoporphyrin is bound to a single iron atom in its Fe3 plus state. Does the metal remains in the Fe3 plus state in case of binding with hemoglobin or myoglobin? Because protoporphyrin means they are talking about the organic moiety that is present in case of hemoglobin and myoglobin. In that case, we all know the metal that is present there is Fe2 plus. It is present at its 2 plus state, not its Fe3 plus state. So this statement is obviously a false statement. Next comes, heme consists of a complex organic ring structure called protoporphyrin. Absolutely, heme consists of a metal ion Fe2 plus and an organic ring structure called protoporphyrin. So this statement is true. Next is, iron atom has 6 coordination bonds. Absolutely, I will show you the structure in a moment. Iron atom does has 6 coordinate bonds. And the fourth option is, so this is also a true statement. Fourth option is, heme is found in a number of oxygen transporting proteins. Yes, it is found in number of oxygen transporting proteins. So the only false statement that was there is the first one, the Fe3 plus state. Now this, what I was talking about, iron atom has 6 coordination bond. If you look here, this is the structure of heme. So this outside you have your protoporphyrin. These are the 4 nitrogen atoms of those porphyrin rings. Now here sits the Fe2 plus. So this is the plane. If you look at it, this is one your plane. This oxygen molecule is sitting actually above the plane, and this pyrrole moiety is actually sitting at below the plane. Okay, so this is the structure of a normal heme. Now if you look at the coordination of Fe2 plus, this is 1, 2, 3, 4, 5 and 6. It forms 6 coordination bonds overall. Okay, so this is already known to you. This question all of you do know, that adult hemoglobin is a tetramer consisting of how many units? It has 2 alpha units and 2 beta units. So that is why it is called alpha to beta 2. Again, this is, I think you all will be able to answer this. This says, this is the hexapeptide that you are treating with CNBR. What are the fragments that you are expecting from the treatment? This is your question. So what does CNBR does? If we do remember, CNBR breaks after methionine residue. I am saying this again and again. So you will get your cleavage from here. So you will end up with KWGM, sorry, KW, one second, KWGM plus LA. Now this particular thing will form KWG lactone. After it breakdowns at the methionine site, it then will form a lactone and we will end up with KWG lactone. This M will now be removed and end up with the lactone. So now you will have KWG lactone plus LA. So the correct option for this should be option number B, KWG lactone and LA. Okay, we already have seen this. The slope of a hill plot for myoglobin does what? whereas there, sorry, for hemoglobin does what, and what does it do for myoglobin. So this is your hill plot here. We already know that this blue one is myoglobin, where the slope stays constant for all partial pressure. So this option for myoglobin, it is constant for all partial pressure, and in case of hemoglobin the slope drastically decrease. At higher pressure you can see this is your slope, but at lower pressure this is your slope. So it decreases drastically. Okay, so this was your slope at high partial pressure. This line is the slope at low partial pressure. So if I draw like this, at high partial pressure it look like this, at low partial pressure it looks like this. So the slope at low partial pressure for hemoglobin decreases. So this was the correct option. Okay, so this was the last question for these slides. We'll go to the week 7 lecture materials, but before that let us take a 5-minute break, and I will be say back at 7:50, and we will still have 10 minutes left. We'll discuss some questions from week 7 and then we'll end the session. Okay, just we'll be back at 7:50. Okay, so let us begin with the session again. Let me. The screen is already shared, I guess. Yes, I think it is visible for me. I hope it is for all of you. Okay, 7. Fine. So let us start with the lecture materials that you have learned in your week 7 lectures. Okay. So only week 7 lectures I think had 3 parts. So these questions are mostly about the first 2 parts of the lectures. The third part we will discuss in the next live session. So let us start with the first question. It states, micelles of fatty acids in water are organized such that the M face the solvent and N are directed toward the interior, and these are your options. Since since we do not have much time, I will directly come to the sorry, I will directly come to the solutions of this question. So you have your fatty acids. How does fatty acids looks? So you generally have a long chain, a hydrophobic chain, and at the end you have your polar head groups at COO minus or carboxyl. So yes, whenever you will put something like this in water, obviously it will try to orient itself as such that the hydrophilic parts will be in contact with the water molecules and hydrophobic parts will remain in the interiors, obviously. So in this question it states M phase the solvent whenever you put them in water. So if M phase is water, then it must be hydrophilic head parts of fatty acids, and N is directed toward the interior when you putting them in water. That means N is the hydrophobic tails that are trying to stay away from the water molecules. Yes, absolutely right, all of you. So correct option for this should be option number A. Moving to the next one, it is saying sphingolipids are derivatives of the lipid sphingosine, which has a long hydrocarbon tail and a polar domain that includes an amino group. Glycosphingolipids, a derivative of sphingolipids, is a combination of what? So these are your options. So you already know sphingolipids is that where the backbone is sphingosine. And what does sphingosine have? It generally, sphingosine itself has a hydrophobic side chain. It does have a NH3 plus which can attach with different amino acids, and there again you have a OH group that you can further functionalize. So what will you call glycosphingolipids? I have one answer I can see. Yes. So yes, absolutely the correct option should be option number B, that is ceramides with one or more sugar residues. So how it is coming? So we have this as our sphingosine. What we will do next? We can attach a fatty acid here. After you attach a fatty acid, we will call them ceramides. Okay, this you need to remember. Whenever you block the NH2 side or NH3 plus side of sphingosine with a fatty acid, we will call them sorry, one second. We will call them ceramides. Now after that we still have one OH group left which we can further conjugate with other molecules. If this OH group conjugates with a monosaccharide, we will call them carboside, and if they attach with, let me write it down here, with a complex or oligosaccharides, then we will call them ganglioside. So this is the question. So obviously in case of glycosphingolipids, from the name itself you can identify that there is a glyco residue. Whenever you see a glyco residue, that means there is some sugar molecules involved. So the sugar molecules have to be incorporated. Sugar means these monosaccharides or complex or different oligosaccharides. They have to be incorporated in this OH site. Now before going to that, this particular site, before you attach any sugar moiety to this OH group, you have to first block this NH2 site with fatty acid. That means first the formation of ceramides will happen, then it will react with any kind of sugar moiety and will result in glycosphingolipids. So the correct option should be ceramides with one or more sugar residues. We do not know that whether there is only one single monosaccharide or there is a complex oligosaccharide. So based on that we can further classify them, but we still don't know them. So altogether we can call them just simply glycosphingolipids. That means a sugar moiety is attached. Okay, moving to the next one. It is a very easy one. I just quickly show that. So they are saying, what is the molecular formula of cholesterol? Nothing else. So this is a big question. If you go through all of this, you will end up with the ultimate question is, what is the molecular formula for cholesterol? Now this is the structure of cholesterol. Now what do you need to do? First of all, this is the basic steroid units that you need to remember. This is your all or the hydroxyl group, for which we call them sterol, and this is the hydrophobic R group that is attached here. This is your normal steroid unit. So you know the structure of cholesterol. In case of cholesterol, the R group is this. If you know this structure, you just have to transform it to the molecular formula, and you will end up with C27H45OH. So this part is your sterol, this part is your sterol. That formula is C17H228 oxygen. Now that you are adding the other molecules, that is ending up with C27H45OH. So this was an easy question. You can just look at the structure and then can write down the formula. I will not discuss much about it. Okay, this is a good question. It states, according to the fluid mosaic model of membrane structures, protein of the membranes are mostly staying in what condition? Okay. So if you do, I don't know whether I have the model for fluid mosaic model here. So these are the 4 options. I think I do. Yes. So this is your fluid mosaic model. In case of that, they are asking how the proteins generally remains. Let us look at the options first. First option says the protein molecule spread in a continuous layer over the inner and outer surface of the membrane. So what is this membrane? This is a protein bilipid layer. So this is your membrane structure, where this is your hydrophilic head groups and this portion is your hydrophobic tail groups, right? So this portion is hydrophobic, if we do write down, and these portions are, this round portion are hydrophilic. In the first option, they are saying that the proteins are spreaded on the surface. That means that the proteins remains uniformly distributed at the outer surface and as well as the inner surface as well. If this happens, ultimately will it result in any kind of favorable transport or anything? Because we all know we need the protein molecules so that we can generate some channels in these phospholipids or the bilayers, so that from outside different ions or say suppose some hydrophilic molecules can pass through these membranes and enter our cells. Now if the proteins are just laying on the surface and are not forming an connection through this bilipid layer, then no transport will occur ultimately. So we will have no advantage. So the first option is absolutely wrong. Coming to the next one is saying the proteins are confined to the hydrophobic core of the membrane. So this is your hydrophobic core. Again, if the proteins are only staying at the hydrophobic core, that means again we don't have any connection to go through the bilipid layers of cells. That means again no transport will occur in our cell. So we will no longer live, we will no longer function. So again this statement is absolutely wrong. If it does only stay in the hydrophobic part, it will not serve the purpose. Next is embedded in the lipid layer. Absolutely yes. These proteins always remain embedded in the protein layers. They are transmembrane proteins. That's why they are called as transmembrane. That is, they both have hydrophobic parts and hydrophilic parts. And whenever, say suppose, whenever they are entering this phospholipid layer, they are creating some, suppose alpha helix is interacting here, they are creating some kind of channel through which the ions and other hydrophilic molecules can, or hydrophobic molecules can pass through. So this is their role. They have to be embedded in the lipid bilayer and act as a transmembrane protein. So yes, our option number C was correct. And the last one is randomly oriented in the membrane with no fixed inside outside polarity. That is absolutely rubbish, because if it does not have any fixed inside outside polarity, it will never be able to interact with the bilayer itself, because we all know the outer side it has to be non-polar and the inner side outer side it has to be polar and in the inner side it has to be non-polar. But if it is randomly oriented, then it will not be able to interact with the bilayer at all. So again, the random orientation part of polarity is absolutely wrong. So the correct option for this was only option C. Okay, we will only do this last question and the rest of the question we will take in the next session. So the next one states a fatty acid designated as 20 is dash, you have to fill it, while one that is designated as 23 delta 5811 is dash. You have to identify it, what are they. Let us come. So first we need to understand what does these numbers mean. So we already know in case of fatty acid we can designate them as delta model. There is two models, one is delta designation and another one is N designation. One is delta designation and another one is N designation. in both the cases, whenever you first write down the number, in both the cases, the very first number means the number of carbon atoms in the chain, right? Throughout the chain length, how many carbon atoms are there, that is it represents. So there are 20 carbon atoms, we already know. Both in case of delta and n, it will be same. What does this next number means? After that, after this notation, if we write 3, what does this 3 denote? In one case it is written as 20 and in another case it is written as 23. So in both delta model and n model, this second number means how many double bonds are present in your fatty acid system. Okay, that means in case of 23 we have 3 double bonds, but in case of this amino acid where it is designated as 20 we have 0 double bonds. That means 20 does not, this fatty acid does not have any double bonds in the side long hydrophobic chain. That means it is completely saturated. If there is no double bond, that means it is completely saturated side chain. So this option should be saturated. But the second one, that is 23, where we have 3 double bonds, that means there will be 3 double bonds attached in some manner. I am just attaching it randomly here. So there is unsaturation in the side chain. So this particular one should be unsaturated. This one is unsaturated. And what does this 5, 8, 11, delta 5, 8, 11 means? In case of delta model, obviously from delta it is obvious that this is following delta model. So delta designation 5, 8, 11 means the position of the double bonds. It is stating us that the first double bond exists between C5 and C6. The second double bond exists between C8 and C9, and the third double bond exists between C11 and C12. So if we have to draw the structure, then we will start from COOH and then continue this side chain till 20. Okay, I am not continuing it. Till 20 carbon, we will start marking from 1, 2, 3, 4 like that, and we will continue till 20, and then at last we will place the double bond connecting the carbon atom. Say this is, if this is 5 and 6, then one double bond will be placed here. Similarly, another next double bond will be placed between 8 and 9, and the last one between 11 and 12. With this, let us finish this session and continue this type of question in the next class. Okay. Thank you all for listening. All can leave now. Thank you.